Welcome to this tutorial on enumerating microorganisms using the pour plate method. This method is commonly used to quantify the number of viable microorganisms in a sample, expressed as colony-forming units per gram or milliliter. Let's walk through the procedure step by step. Equipment and materials. Before we begin, you will need the following equipment and materials: sterile Petri dishes, sterile pipettes, sterile dilution tubes, sterile media for the specific test, sterile diluent such as saline, 0.1% peptone water or Butterfield's phosphate buffer BPW, etc. To test sample, an incubator set to the required temperature, usually 35 degrees Celsius for bacterial plates and 25 degrees for fungal plates, and a colony counter. Now let's start our analysis. Weigh the desired amount of dehydrated media as according to manufacturer's description. Measure and add the required volume of purified water. It could be distilled water, osmosis water, or any form of purified water. Add the water in bits and swirl to mix uniformly with the dehydrated media. Heat and stir the media where necessary to ensure complete dissolution of the agar component. You can as well use a microwave in place of a heating magnetic stir. After the media dissolves, transfer into the final container, which in our case is a media bottle. Sterilize the media in an autoclave machine as directed by the manufacturer at the required temperature and pressure. After autoclaving, temper the media in a water bath set to about 45 to 50 degrees Celsius. Disinfect your work surface with 70% alcohol and wipe it down with cotton wool or tissue. Prepare your sample by dissolving X grams or milliliters of sample in 9x volume of sterile diluent. Mix the first dilution by swirling and allowing microorganisms to come into solution. To pet 1 milliliter of your first dilution into a tube of 9 milliliters of sterile diluent to make up your second dilution. To pet 1 milliliter of your second dilution into a tube of 9 milliliters of sterile diluent to make up your third dilution. And do that for other subsequent dilutions you may need. For the pour plate method, which is the focus of this video, transfer a known volume of your inoculum into a sterile Petri dish which has already been labeled with your sample ID and the dilution factor being inoculated. Pour enough media to cover the plate and swirl to mix uniformly. Allow the plates to solidify at room temperature, after which you can incubate. Incubate bacterial plates at the required temperature of 35 degrees Celsius, or otherwise as stated by your method, ensuring that the plates are held upside down and not stacked so high. After the period of incubation, count the colonies as observed on the plate and record the number. Go on to calculate the colony-forming units as the number of colonies counted, multiply by the dilution factor and divide your answer by the volume of inoculum pipetted or used in the Petri dish during the inoculation.